Okay, so today I'm going to be talking to you about chromosomal mutations. Now, these are different from the gene mutations that you learned about in the earlier unit. If you haven't watched the gene mutation video, please watch that first and complete those assignments before you do the assignments associated with this video. So, chromosomal mutations are mutations that affect either the number of chromosomes a person has or the structure of the chromosomes that a person has. Now, chromosomal mutations are different from gene mutations because they affect many genes. They may affect 100 genes or 1,000 genes, whereas a gene mutation, like a substitution or a point mutation, affects just one gene and therefore one protein. Chromosomal mutations will affect many genes and therefore many proteins, hundreds or even thousands. So we're going to look at the two different categories of chromosomal mutations a little more closely. First, we're going to look at changes to chromosome number, and then we'll look at changes to chromosome structure. So ultimately, changes to chromosome number are caused by something called non-disjunction. And ultimately, non-disjunction just means the failure of chromosomes to separate. Now, this failure to separate occurs during anaphase of either mitosis or meiosis. If it happens during mitosis, then it's going to be passed on to, the mutation will be passed on to body cells. If this happens in meiosis, which is the most common scenario, it's going to be passed on to the offspring because it's going to produce gametes or sex cells like sperm and egg that have the wrong number of chromosomes. So if non-disjunction occurs during meiosis, some of the sex cells or gametes will have too few chromosomes and others will have too many chromosomes. And should those sperm or egg be fertilized, then you're going to end up having an individual with the wrong number of chromosomes. So make sure that you know a couple of things from this slide. Make sure that you know the term non-disjunction, and make sure that you know that chromosomal mutations affect the number and structure of chromosomes, and they cause cells to have too few chromosomes or too many chromosomes. So those are the key things to take away from this slide. All right, so now let's look a little more closely at our changes to chromosome number. So how would we even know that somebody has the wrong number of chromosomes? So when we suspect that a person might have a chromosome number disorder, we take a picture of their cells during metaphase, and we then put the chromosomes into pairs based upon their size and their banding patterns. And we'll talk more about how to interpret karyotypes in another video lesson later on. So what you want to look at when you see somebody's karyotype is, okay, a normal person would have 2 chromosomes at every numbered position. So notice that there are 2 chromosomes above number 1, there are 2 chromosomes above number 8, there are 2 chromosomes above number 17. And then you would look at the last pair of chromosomes, and you would want to make sure that the person either has 2 X's, indicating that this person is a female, or they have an X and a Y, indicating that the person is a male. So if you added it all up, a normal person would have a total of 46 chromosomes. So if a person has a chromosome number disorder, they're going to have a number more than 46 or less than 46 when you look at their karyotype. So there are 2 types of chromosome number disorders that are common in humans. Those are called trisomy and monosomy. So trisomy, what do we mean when we say trisomy? Well, tri means 3. So in trisomy, a person has 3 chromosomes at one of those numbered positions or at the sex chromosomes instead of the usual 2. So this is because there's 1 extra chromosome in one of the gametes that formed that individual. So in trisomy, a person will have 1 extra chromosome, or a total of 47 chromosomes. And a couple of examples are Down syndrome, which a person has an extra copy of chromosome 21, and Klinefelter syndrome, which is a disorder that affects only males, and that male would have an extra X chromosome. So instead of just being XY, he would be XXY. And again, that's caused by non-disjunction. Now, monosomy is also caused by non-disjunction, but in this scenario, the individual is inheriting or getting the cell that is missing a chromosome. So this person is going to have one too few chromosomes, or 45 total chromosomes instead of the regular 46. An example of this in humans is Turner syndrome. Turner syndrome only affects females, and this is a person who has only one X chromosome. So there's an X, and then there's nothing else. There's not an extra X, so they're not paired X's, and there's not a Y. So these people are considered female because they do not have a Y chromosome, even though they are missing one of the sex chromosomes. So the monosomy, again, is one missing chromosome. So you can also think of it as only having one chromosome at maybe a numbered position or at one of the sex chromosomes. All right, our last type of chromosome number disorder is called polyploidy. Now, polyploidy affects plants, not humans. Now, if polyploidy occurred in humans, it would be a fatal mutation, and the embryo would fail to form, and there would be a miscarriage. So in polyploidy, you get a whole set of chromosomes that fail to separate. So that means that if a plant had 16 sets of chromosomes, all 16 sets fail to separate, so that you get extra copies of every single chromosome. So this happens, again, just in plants, and a lot of the plants that we use for fruit, like strawberries, for example, and citrus, like oranges, are polyploidy. And that means that they have extra sets of chromosomes. Think strawberries are octoploid, so that means they have 8 sets of chromosomes per cell. And again, this is fatal in humans. So humans, trisomy and monosomy are the common types of chromosome number disorders. Polyploidy is a special one to plants only. Now, when we're talking about these number disorders, we are going to practice reading some karyotypes in another lesson, and you're going to have to pick out if they have a trisomy or monosomy, so make sure you've written down these words so that you have those available when you do that. So the next type of chromosome disorder that I want to talk to you about are chromosome structure disorders. Now, a structure disorder does not change the number of chromosomes that a person has. It just affects the arrangement of the segments or the bands of those chromosomes. And there are 4 different types of chromosome structure disorders that you need to be familiar with. The first one is a translocation. So a location means where something is. Trans means across. So that basically it's going to move from one place to another. And this rearranges our chromosomes by breaking off one and reattaching it to another chromosome that's completely different from it. This is the only chromosome structure disorder that we're going to discuss that involves 2 different chromosomes. So that's something that you can use to help you distinguish it from the other 3. So there are 2 different chromosomes. Parts of the chromosomes break off and reattach to another chromosome. Now this could be one chromosome breaking off a segment and attaching to a different one, or both of the chromosomes can break off pieces and then basically trade places with each other, and that's called a reciprocal translocation. The next type of chromosome structure disorder is called a duplication. So duplicate means to copy. So you're going to see that a segment of the chromosome is going to be repeated. The third type is the deletion, and delete means you take something away. So a segment or segments of a chromosome will be removed, and therefore you're removing all of those genes along that chromosome. Our fourth type is an inversion. So an inversion, think in reverse. So usually around the centromere, the segments of the chromosomes flip so that they're inserted backwards and the gene sequence is reversed. And this is a problem for the cell when it starts to try to transcribe a gene, for example. Everything will be messed up. So remember, there are 4 chromosome structure disorders: a translocation, a duplication, a deletion, and an inversion. Now let's take a look at the pictures of each of these. All right, the first picture that I want to show you, first let me say this. when you're looking at the pictures, one side of the diagrams will be the original unmutated chromosome. So everything on this side are the original chromosomes. And then on the other side will be the mutated version, and you have to decide what type of mutation has occurred. Now, for this, I've given you the names of the mutations on the pictures. Later on, you're going to have to figure it out on your own as parts of your grade. So think of it kind of like as the original as the before and the mutation as the after. So you have to compare the original to the mutated sequence and figure out how they're different from each other to determine the type of mutation. So if you look at the way that this one is arranged, this chromosome has a total of 6 segments. Segments A, B, and C are in front of this circular structure here. That circular structure is the centromere. And segments D, E, and F are after it. Now let's look at the mutated sequence. It's A, C, D, E, F. So if you compare the two, you'll notice that the mutated sequence is missing segment B. Therefore, this is a deletion. And all of those genes that are on segment B are now gone, and those proteins cannot be made. Now let's look at our second example. Again, we have segments A, B, and C in front of the centromere, segments D, E, and F after the centromere. Now, how is the mutated sequence different? Now, the mutated sequence is A, B, B, C in front of the centromere and D, E, F afterward. So notice there are 2 segment B's, so that means part of it has been repeated or copied, so it's a duplication. Now on this diagram, you're seeing that the chromosome's kind of twisted. It's still just one chromosome. I don't want you to rely on that to be how you pick out this type of mutation. Instead, just imagine that it's straight, just like the one in front of it. Notice that you have segments A, B, and C in front of the centromere, and segments D, E, and F still after the centromere. So how is it different? We still have segment A first, but now it's ED, CB, and F. All of the segments are still there, just that segments B and C are flipped with segments D and E around the centromere. So when you have all the segments but a sequence is in reverse order, it's called an inversion. So everything's still there, it's just in the reverse order. Our last example is a translocation. Now, translocations should be the easiest to pick out because you have 1, 2 chromosomes, whereas everything else above it had just 1 chromosome. So that's your clue. If you see 2 chromosomes, automatically know it's a translocation. So the translocation, you're going to see that part of one chromosome breaks off and reattaches to another chromosome. Now, this could involve a reciprocal translocation where both chromosomes have segments breaking off, which we do see this happening in this example, and trading places. So our original one, A, B, C, D, E, F, now has basically the D, E, and F has traded places with the J, K, and L on the other chromosome. So that's a translocation. And remember, if you see 2 chromosomes, you automatically know it's a translocation. You don't even have to look at anything else. Now, I've talked a lot about mutations, and it sounds like, oh my gosh, they're happening all the time, but in reality, they're not. First of all, we have a really low mutation rate when we replicate our DNA in the first place. We only make a mistake like once every billion nucleotide pairs that we copy. And most of the time, our cells are going to check the replicated DNA and actually catch those mistakes before they can be passed on to any new cells. Remember, we talked about those checkpoints when we talked about the cell cycle in cancer, and that was the job of some of those things, to prevent those cells from moving forward if there was a mistake. So just remember that we've got these proofreading enzymes and repair enzymes that fix those mistakes, which occur very infrequently in the first place. Now, our rate of mutation can increase when we are exposed to substances that can cause mutations, and substances that cause mutations are called mutagens. So mutagens can include things that can cause cancer, like UV radiation and cigarettes, but they can also include things like radioactive material and x-rays that can just cause birth defects. So not all mutagens cause cancer, but they can. Some mutagens cause birth defects, and those can lead to those changes in your chromosome number and chromosome structure. So just make sure you remember that a mutagen is anything that can cause a mutation, and you already know some examples from when we did our unit on cancer. All right, and lastly, I just want you to remember mutations are not always bad. Mutations can be good or they can be neutral. So when we talk about mutations, don't think everything is doom and gloom. Sometimes mutations are beneficial, meaning that the organism that has those mutations end up being better suited to whatever environment they're living in, and ultimately over time those traits become more common because those individuals produce more offspring, and that kind of will lead us into our next unit on natural selection, which is how organisms change over time or evolve. So just remember, mutations aren't always bad. They can be good or they can be neutral, meaning they have no effect either way on the organism. Right. Once you're finished watching this video, make sure you watch the karyotype video on how to read and interpret karyotypes and practice that, and then you can do the daily grade on chromosomal mutations and karyotypes.